Welcome back to Unit 3, Video Lesson 1, Part 2. Again, this is optional unless you've missed class. In this case, we're going to pick up with base pairing. And it's important to remember in terms of base pairing that a purine, whether you're DNA or RNA, a purine always pairs with pyrimidine. So what you get, you always get that purine being the two-ring structure is always going to pair with the single ring structure. So at any place in the DNA ring, you should only have a total, or DNA strand, you should only have a total of 3 rings across. And in DNA, it's A double bonds, 2 hydrogen bonds to a thymine, and G 3 hydrogen bonds to a cytosine. And this is a stable situation, the long-term form. In RNA, it doesn't typically form 2 sides of a chain, but when we're actually building it from DNA, an A will always pair with a U, and a G will always pair with a C. Okay, this is just temporary in most cases, but there are some situations where RNA folds back on itself and these types of bond pairings also occurs. So now we're going to look at how the chain structure forms, and I'm going to have to write fairly small, so feel free to zoom in when you're watching. We're going to start with a single nucleotide of DNA. We're going to represent the phosphate, the base. That phosphate is going to attach to the 5' sugar of the nucleotide. Go straight across. And we're going to have the next, pick a base first. Let's finish this nucleotide. Usually we just draw it as a rectangle, not the more complicated form. And we could pick a base. Let's just start with A. Okay. And because it's DNA and it's missing an oxygen, we'll indicate that here with just the H. And then the next phosphate is going to attach at the 3' carbon. Pick another base. This time I'll pick T. And then we draw the next one. Another base. C. One more time. See how the phosphate's attaching to the 5' carbon, and then when we bring in the next nucleotide, it attaches to the 3' carbon. That's why we say that DNA grows 5' to 3'. And what base have we left out so far? That's right, G. I'm sorry, I'm going to have to go over into the RNA space a little bit, so we'll just draw a new line here to finish this drawing. Now let's go back and add in those H's so we're clear that we're talking about deoxyribose and DNA. Let's indicate that this chain grows in this direction, what we say 5' to 3'. Now DNA is typically double-stranded, so all those bases are going to pair up with a complementary base on the other side. Complementary means they fit together perfectly, not that they're identical. So in this case, if you have your page, you notice how this is coming across on an angle, and I'm just going to draw this in another color, how this is coming sort of across on the angle like this. Well, if we were just to add another chain, 5' to 3', it would be coming across on an angle like that, and it'd be very difficult for the top ends to get together, and that would be ineffective. It wouldn't work well. So instead, I'm just going to try and erase this line. I'm not sure I can. Yes, good. Instead, it flips. Instead of growing in the same direction, 5' to 3', top to bottom, it's going to grow 5' to 3' though. It's going to start at the opposite end. What I recommend now is you just change your page. I'm going to try and spin my computer around and write on it upside down. We'll see how well that works. I think we're okay. So if we start down at this bottom, hopefully you've changed your page around now, upside down. Again, get the phosphate attached to the sugar, attached to the base. And what's going to complement with the G? That's right. A C, how many bonds? Very good, 3. Then off that 3' carbon, we're going to attach the next nucleotide. See how now they're, because it's what we say anti-parallel, it's able to sort of reach back to the other base. That's going to be a G to complement the C. Do the next one. What base are we going to complement the T with? That's right, A. How many bonds? 2's correct. One more. What's going to complement the A? T, correct. How many bonds? 2. Okay, and just because, again, we want to stress the concept that this is DNA, deoxyribose, so we're going to put the hydrogens in. And we're going to indicate how it grew, 5' to 3'. And now I can flip my page back around, and so can you. And what we get is this so-called 5' to 3' on one side, and coming up the opposite direction is the 5' to 3'. That's what we call, that's what we call anti-parallel. If they were both running in the same direction, that would be parallel. Okay, so anti-parallel is that fact that it has to flip just so it can fit together perfectly in three-dimensional space. Now we're going to take a little closer look at this. I know it's getting really messy, but if you've drawn it and followed along, it shouldn't be too bad. We are going to show how we have a sugar phosphate backbone. Let's choose this color. We call this, in DNA, the sugar phosphate backbone. Or, because there's actually 2 of them in DNA, and it kind of looks like a ladder, we call it or sugar phosphate rails. The rails are the sides of the ladder. So just in case you forget, just like the rail on a stair is the side that you hold on to, whereas this part you're supposed to put your feet on, those are the rungs. Fortunately, many of us grab onto the rungs with our hands, but that's not the intent. Okay, so what are the rungs made up of in this case? We'll change the color here. Let's go yellow. A little bit difficult to see, but the rungs are down the middle, and the rungs equal hydrogen bonded bases. Okay, I won't choose yellow again. I'm not sure that was very effective. So the rungs are the yellow part. Okay, and that is the structure of DNA, except for due to the way the chemical bonds are, it twists into what we are probably more familiar with, the double helix. I'm just going to pause there. I will come back in part 3 and we'll talk about the structure of RNA. I'm going to clean it up though. I'm going to erase this section so it's nice and clean when we look at RNA. Okay, if you have any questions about this, you can come see me in class. Okay, I will see you in a second over in part 3.